Hello and welcome to today's Low Trophic Life Webinar. Today's presenter is Stef Claessens, who is a master student from University of Algarve in Portugal. His title is Does Mussel meal improve growth in the white-legged shrimp? But first, I would like to just talk a little bit about Aquavitae. Aquavitae aims to increase the aquaculture production in and around the Atlantic Ocean by developing new species, processes, and products. The focus of the project is to placed on low trophic species such as algae, sea urchins, and shellfish. The project will work to create real and meaningful collaboration between researchers, industry, and other aquaculture stakeholders in the Atlantic area. I will be linking to the Aquavitae webpage where you can go for further information. You can also follow us on Facebook, Twitter, Instagram, LinkedIn, and of course our YouTube channel, where this recording will go afterwards. But back to you, Stef. Before we begin the webinar, I know you're going to make your own introduction, but why this research topic? Why this? All things.
Yeah, first of all, I like nutrition, of course, and nutrition aquaculture, I believe, is one of the most important things in aquaculture. And when I noticed this project where we use low trophic species into aquaculture feeds, I like it. And also because the practical part is in Brazil, so new experiences. I already been once to Brazil. I was working there also 8 years ago on the fish farm. So yeah, overall, I'm very happy with this thesis project.
That's very good. So you can just go ahead and make your presentation now if you want, and then we will have the Q&A session afterwards.
Okay, perfect. You can see the presentation, right?
That looks good.
Okay, so hello everybody. My name is Stef Claessens, and I am a master student from the University of Algarve from the south of Portugal, city called Faro. Originally, I'm from the Netherlands, and today I will tell you about my thesis project. This thesis project is part of the Aquavitae student exchange project, where I went to Brazil, to the south of Brazil, to Florianópolis, to test if mussel meal can be used as a potential ingredient for a diet for the white-leg shrimp, and also to see if it improves growth. And this experiment I did at the Marine Shrimp Lab from the University of Santa Catarina. First, I will give you a short introduction and tell you what we have done and why we have done this research. After, I will tell you the method, is how we have done the experiment. And then I will tell the results, conclusion, and at the end, I will try to answer all your questions. So why we have done this experiment? First of all, for the people that are new in aquaculture, new in aquaculture, the fish meal and fish oils that are used in aquaculture feeds are coming from the fish from the ocean. So there's also one of the reasons that the national fishing stocks are decreasing. And when the stocks are decreasing, the price of the fish meal and fish oil are increasing. So that's why researchers are looking for potential ingredients. And one of these potential ingredients can be mussel meal. Mussel meal is made from the meat from the mussel, and it's rich in protein, lipids, also the unsaturated fatty acids like omega-3 and 6, for example, minerals and vitamins. And besides this, mussels are a low trophic species. So this means that they are low in the food chain. And this means that they minimize losses in between trophics to build up animal proteins. And I have here a picture to clarify this a little bit for the people that don't know. Like in the first trophic, you have the lower part, you have the phytoplankton like the algae or plants. And the second trophic, you have the filter feeders, so shellfish or fish that eat these plants. And then you have the fish that eat these animals. And on the top, you have the top predators, for example, a tuna or a shark that eat these fish again. And in between these trophics, you have energy losses, like around 90%. And this because of the animals has to grow, they have to swim. So, for example, if you want to have a pound of tuna meat, you need in the beginning a biomass of 1,000 pounds of plant or algae. But a mussel that is like in the second trophic, it's like a filter feeder. It can make 100 pound of biomass from the 1,000 pound of phytoplankton in this case. So in this case, like the mussels are way more efficient and way more sustainable than, for example, a tuna. In our experiment, we have used the Litopenaeus vannamei. This is a shrimp species, Litopenaeus vannamei. Also, common name is the whiteleg shrimp. It's by far the most farmed shrimp worldwide because of the high growth, high tolerance for salinity, oxygen. You can stock them in high densities. They have a low protein requirement in the diets. And they are native. They are native to tropical areas. We have a temperature all year above 20 degrees. But because of the high value of these species, farming of these shrimps are expanded to the subtropical areas. For example, south of Brazil. And this on the photo, you see the Litopenaeus vannamei. It's on the photo. It looks a little bit bluish. But that's because they adapt the color from the tanks. So that's why this one is a little bit bluish, because we have blue tanks. For the mussel meal, we use the species Perna perna. It's the most farmed mussel species from the southern Brazil, especially in the state Santa Catarina. And at the end, we also tested the Coldshock resistance because the farming of these species are expanded to the subtropical areas. A temperature is like one of the major concerns during farming these shrimps, because they reduce growth and they are more sensitive for diseases, for example. So that's why we also tested if a mussel meal have positive benefits for the cold resistance. So first, before we started the experiment, we had to make the mussel meal, and this we did by using the protocols that were developed by our partners from SISKI. It's another case study from the Aquavita project, and these are the steps. First, we had to cook the mussel for around about 10 minutes when they open the shell and take out the mussel meat. Then we put them in a dry oven for 48 hours at 70 degrees Celsius, and we use this temperature so that the proteins and lipids didn't alter, so they stay intact. Then we miced the dry mussel into smaller parts, and then we sieve it till everything changes to a powder from less than 600 mu. After making the mussel meal, we had to formulate the diets. We used 5 different diets, 5 different treatments. Like the control is without mussel meal, and then we put 1, 2, 3, or 4% of mussel meal in the diet. And we had to take into account that mussel meal is very rich in protein and lipids. Protein is about 55% of dry weight, and lipids almost 15% of dry weight. So to compensate this, we took a little bit out the soybean meal, fish meal, poultry meal. So we keep the protein, lipid, and energy levels for all the diets the same. And this is very important, because then you can really compare the different diets with each other. Then the experimental setup. We use 5 different treatments with 4 replicates. So in total, we had 20 tanks of 400 liters, and we keep them for 8 weeks, the shrimps. Each tank we stocked with 40 shrimps with an additional weight of 3.5 grams, around 3.5 grams. We fed them 4 times a day, every day, and the water camp temperature was around 28.5 degrees, dissolved oxygen we keep between 5.9 and 6.5 milligrams per liter. We had a day-night regime of 12 hours light and 12 hours dark. And every day we changed the water 100% to keep the water parameters suitable for rearing the shrimps. So to keep the nitrate, ammonia, etc. right. Then to check the Coldshock resistance, we did a Coldshock treatment, and this we did by putting the shrimps for 1 hour at 10.9 degrees, and then put them back at 28.5 degrees, and we were measuring the mortality for the next 24 hours. These are the most important growth results, the final weight and the FCR. The shrimps, they started at 3.5 grams, and after 8 weeks, we had the final weight, the average final weight. So, for example, for the control, it was like 18.5, around 18.5, and for the 2% muscle meal diet, it was 20.4, and the letters that you see, the A and the B, this means that the 1 and the 2% muscle meal diets, they had a significant better growth than the control 3 and 4%. And on the right, you have the FCR, the feed conversion ratio. This means like how many kilograms of feed you need for 1 kilogram of shrimp. So the lower, the better. And also here we see, if you look to the letters, you see that the 1 and the 2% muscle meal diets had a significant lower FCR than the control 3 and 4%. So, for example, if you look to the 2% of muscle meal diet, you need 1.16 kilograms of feed to produce 1 kilo of shrimp. Then the survival. The survival was in all treatments above 94% after 8 weeks. So that's perfect, almost nothing died. And we didn't see any difference in survival between the treatments. Then the Coldshock results. It's very important that the control had around 50% of survival, or 50% of mortality. So you can compare it with the other treatments. But in our case, in all 5 treatments, with or without muscle meal, we had almost the same survival. So the muscle meal didn't have any positive benefits on the Coldshock resistance. Already the conclusions. If you look to the growth, health, and survival of the shrimps, I think we can conclude that muscle meal can be used as a potential ingredient in diets of the whiteleg shrimp. And the shrimps that were fed with the 1 and the 2% muscle meal diets were heavier than the control 3 and 4% diets after 8 weeks. They even grow in average 2 grams per shrimp per week. And we were aiming during the experiment at least for 1 gram per week. So the growth was really good. And the best growth we had from the 2% muscle meal diet, and they were 10% more heavier than the control. So by putting only 2% of muscle meal in a diet, the growth was 10% more after 8 weeks. So this is also a very nice result. And this was already my presentation. Thank you for your attention.
Thank you very much, Stef. Before we begin the Q&A session, or while people are typing questions in the chat, I don't see any questions there so far. So I'd just like to ask the first question. Clearly, the results were better with the 1 and 2% muscle meal diet, but I noticed that the 3 and 4% muscle meal diet led to the same growth as in the control groups. Does that indicate that after increasing the percentage of muscle meal into the diet past 2%, it no longer leads to better results? And why is that?
Yeah, that was also because you would think like the more the better, maybe. But it's also very strange for us. It can have like different reasons. one of the reasons what we think can be is maybe because they had the same protein and lipid levels, but the amino acid profile and the lipid profiles, they can vary a little bit. So maybe the 1 and the 2% diets had a better matching lipid and amino acid profile for the needs for the shrimp. This can be, but it's, because the difference were very small, I'm not sure about this. And I also, the other thing can be maybe the oxidation. I went to the Aquavita meeting in Porto, and a guy said, a professor from South Africa said, like, maybe it can also be the oxidation. And that was a pretty good, yeah, it can be because mussel meal are very rich in unsaturated fatty acids, so the long chain fatty acids, and these are also very sensible for oxidation. So when you put more from these in your diets, maybe the oxidation gets more and more. So it doesn't mean the more the better. But to be sure, we don't know yet because we didn't check the amino acid profiles or the lipid profiles, and also we didn't check the oxidation. But it can be the reason for that.
That kind of leads to my next question. As I don't see anything in the chat, what would be your recommendations for what to do next for future experiments? Would that be to look further into this?
Yeah, exactly. I think if you do like to analyze also the feed on lipid profiles or amino acid profiles, so you can really compare per amino acid, for example, then you have like a better view of why the 1 and 2% had better growth. And also the oxidation you can check by the feed. And also it was like very, very interesting or very funny because the shrimps are also very tasty. In my opinion, of course, we took care of them, so I'm not sure if it's like because of this, but it would be also nice if you could do like a taste testing, because if the mussel meal has also a good benefit for taste of the shrimp, yeah, then it also can change a lot of things. So you can, yeah, that will also be a very nice result if that would be.
I see that we have a question from the Aquavitae project coordinator, Philip James. So could you turn on the camera and ask your question?
Yeah, actually, I put it in the chat, but nice talk, Stef. Thank you. And as you know, this is an area close to my heart because I did, when I was a young researcher, I did research on using mussels and spiny lobster. That actually is a follow-up question. You really sort of covered it, Casper, but that there seems to be such a big difference in success of the diets at 2% related to 3%. That was sort of my question as well, why you think that might be. And I have a follow-up question, which is, at the moment, are the mussels produced in Brazil for the local food market, and do they produce more than they like? Is there a surplus supply of mussels that could be used for these feeds?
I'm not sure about this, but I went to the Florianópolis, and you see them everywhere in the ocean. So Santa Catarina is, yeah, I think they farm there the mussels. But because it's a huge country, I'm not sure if they have leftovers. If you asked about this. But,
and any other comment on, you know, as Casper alluded to, there's just such a a difference between 2 and 3% inclusion. You think it may be, I mean, you've sort of explained it, but that was sort of my question as well, really. It was such a striking difference, both in terms of food conversion rates as well as growth.
Yeah, I can get maybe the sheet. Yeah, that's true, but also but it is yeah, we really saw these differences, and then you start thinking why we have these differences, but I cannot say for sure this or that, but that we see the differences, that's for sure. We had also very low differences in between the treatments, so the standard deviations were very low, so we had really significant differences that the 1 and 2% had a better growth and also a better FCR.
Yeah, it's a nice result. It's very good.
Yeah, exactly.
Well, does anybody else have any questions for Stefan? I don't see anything in the chat except for Philip's comments. So feel free to raise your hand if you have anything to ask. I do not see anything coming in. So, oh, we have one from Paula.
Hi. Can you hear me?
Yes, we do.
Okay. I would like whether in the future you would consider analyzing any bioactive properties or compounds in your mussel meal.
If I would be
in the ingredient that you have introduced in your feeds, analyze any possible, because growth could either be related with the bioactive properties of the testing ingredient.
Okay, you fall away a little bit in between, but we also tested. We took out the hemolymph of the shrimp.
Okay.
And we tested them on total white cells and other ones, but these results I still have to analyze.
Okay. 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 Yeah, I was just wondering if there was an effect like that.
Yeah, because we had like pretty good growth results. We took out because of this. We took out the hemolymph. Yeah, I can send it maybe later if analyze the data as well. If you...
I will follow your publications.
Okay, perfect.
I was just wondering what else you would be doing with this.
Okay.
Thank you.
Yes, the floor is open for any further questions. I do not see any, but I'll just give you a minute to a few seconds to write it in the chat or raise your hand. I don't see anything coming in. So if there are no further questions, then I would like to thank you, Stef, for being here today, and of course to wish you good luck with your defense next month.
Thank you.
And I would like to also thank everybody who participated today in the meeting. Of course, we have our next webinar will be at the end of November, where we will have actually a double up session on oysters. So that's something to look forward to. And thank you very much for today.
Thank you.